Hi everyone, and welcome to this lecture on the autonomic nervous system. So let's look at today's lecture topic. So we're going to start off by going back to discuss the overall divisions of the nervous system, and then we're going to focus on the autonomic nervous system, also ANS for short. Within the autonomic nervous system, we have autonomic afferents and autonomic efferents. So we're going to describe those types of fibers. We're then going to jump into the divisions of the ANS, specifically the parasympathetic division, followed by the functions of the sympathetic division. We're also going to look at some specific examples of autonomic control. So we're going to focus on heart rate, respiratory volume, urination, and defecation. We're also going to be looking at the control of the pupil and the lens, but in a later lecture, not today. Then we're going to talk a little bit about autonomic lesions, and we're going to finish off by looking at those levels of control for the autonomic nervous system. So let's start by recapping those subdivisions of the nervous system. So we should all know by now that the nervous system is composed of the central and the peripheral nervous systems. The central being the brain and the spinal cord, and the peripheral consisting of the somatic and autonomic divisions. So let's review that somatic division. So the somatic division of the nervous system is that one that's in charge of controlling and monitoring our external environment, and it's under our conscious awareness. We're conscious whether we're feeling something's touching us, or the information we receive from our senses: vision, hearing, taste, smell. All of this belongs to that somatic division. We can also control voluntary actions of our muscles, skeletal muscles. And the somatic division in the PNS is carried by the 31 pairs of the spinal nerves, if we are in the level of the spinal cord for the body, or the cranial nerves for the structures of the head and neck. So we have somatic motor fibers as well as somatic sensory fibers. So we all know by now that the somatic sensory fibers, they enter via that spinal nerve and via the dorsal root of the spinal nerve to enter into the dorsal horn of that spinal cord, or dorsally into that spinal cord, depending on the pathway. Whereas those efferent fibers, we find their cell body in the ventral horn of the spinal cord, and they leave using that ventral route towards the periphery. So a trick that I gave you earlier in the practicals was this one here: in the door, out the vent. So information comes in via the dorsal roots and out via the ventral roots. But really, we're going to focus on the ANS today. So the ANS, in contrast to the somatic system, controls the internal activities of our organs and glands. So we are not really conscious or aware that these activities are going on all the time in our body. And we have two divisions to the ANS: the sympathetic branch and the parasympathetic branch. And we're going to discuss each of them in detail. So what is the ANS? So as we were saying, it is a largely involuntary, under involuntary control. So we're not aware and we cannot really control it. And it monitors our internal environment, our internal smooth muscles, glands, and organs to maintain homeostasis. So homeostasis is that state in which our internal environment is always in a balance, nice balance. So it's the ideal state to maintain everything nicely and maintained. Okay, so the ANS is influenced rostrally by the hypothalamus, as we're going to discuss later on. So in homeostasis, we always have a dynamic balance between the two autonomic branches: the parasympathetic branch of the ANS and the sympathetic branch of the ANS. So the parasympathetic branch of the ANS is typically referred to as the rest and digest branch. And this branch is typically taking over when we can relax and save energy. The other branch, the sympathetic branch, is typically referred to as the fight or flight system, which prepares the body for stress and it gets it ready to spend energy. Okay, so we can see in this very nice illustration we have a balance here. It can be tilted one way or the other depending on what we need at a specific moment. So if we're allowed to rest, sit on the couch, watch a movie, our parasympathetic nervous system is going to take over. It's going to allow us to digest, to perform those functions that sustain bodily activities, normal bodily functions. But if we're getting ready to run a marathon or we're running from a snake in this case, the body really needs to prepare to spend some energy, get some blood flowing to those skeletal muscles, get some more air into our lungs, get our heart rate up and that heart pumping, getting ready to run away and spend some energy. So let's look at those autonomic fibers, in this case using the example of a slice through the spinal cord. So we have autonomic visceral motor fibers as well as visceral sensory fibers. So those motor fibers are going to be efferent to innervate smooth muscles, cardiac muscle, or glands. And the visceral fibers bring sensory information from receptors in an internal organ. So let's start by looking at those efferent fibers. So the efferent fibers, they have their cell bodies in the lateral gray horn of the spinal cord. And we defined that the lateral gray horn only exists between segments T1 to L2 and S2 to S4 of the spinal cord. Nowhere else in the cord. So that's where we have those cell bodies of the visceral efferent fibers, and they use the ventral root as well to exit the cord to bring about a response in those smooth muscles, cardiac muscles, and glands. The visceral afferent fibers, which bring visceral sensory information from those receptors in the internal organs, they actually follow the path of the somatic sensory fibers as they use this dorsal root of that spinal nerve to enter dorsally into the cord. And they also have some cell bodies in the dorsal root ganglia. So the in the door, out the vent trick still applies for the autonomic divisions. So these autonomic fibers, whether they're afferent or efferent, they only exist within the segmental spinal nerves between T1 and L2 or S2 to S4. And if we're talking about autonomic supply for the head and face structures, those are under specific cranial nerves that we're going to discuss about. Well, we've actually seen these cranial nerves before. So let's expand a little bit upon those autonomic visceral afferent fibers. So in this case, we have an example, the heart, one of our organs. And those afferent fibers, as I was describing, they use the dorsal root to enter into the dorsal aspect of the spinal cord. So they bring sensory information from those organs towards the CNS. They have an important role in telling the CNS about the state of those internal organs, so they really help regulate autonomic function. And they're responsible for referred pain. So let's talk a little bit about referred pain. So what is referred pain? Well, it actually is defined as when there is pain sensations coming from visceral organs, that pain is often perceived in regions of the body surface. So rather than being perceived in those organs themselves, that pain is referred to and perceived to by those areas of the skin. So the brain thinks this pain is coming from those areas of the skin. So a classic example is in situations of myocardial infarction of the heart. That referred pain is referred to the area of the upper chest on the left hand side, as well as the left upper limb. So what's going on? Well, let's have a look into this schematic view on the left hand side. And let's understand that we already talked about how those somatic afferent fibers, let's say from regions of the skin between T1 and T4, those somatic afferent fibers, they have their cell body in the dorsal root ganglion, and they use that dorsal root of the spinal nerve to enter into the cord, correct? But the visceral afferent fibers, those autonomic sensory fibers, let's say coming from the heart, they actually follow the same pathway. So these autonomic afferent fibers coming from the heart is actually innervated by afferent fibers between T1 and T4. Those afferent fibers, they follow the same pathway to enter into that spinal cord. So the exact mechanism for referred pain is not exactly understood. But one theory is that because these somatic afferents and visceral afferent fibers share the same spinal nerves as they enter, as soon as that information starts going up and reaching the brain interprets that information as if it's coming from the regions of the skin. So hand by hand with this theory is the fact that we normally don't really have pain impulses from the viscera, from our organs. It is only until something goes wrong. But the body is often receiving pain impulses from the skin. So it is another reason or a possible explanation as to why the brain interprets the information as if it's coming from those regions of the skin innervated by the same segmental spinal nerves. Okay, so there is other kinds of referred pain from other organs, but this is the classic example, pain from the heart being referred to this region of the upper chest and left upper limb. So let's talk about those visceral efferents now. So the efferent fibers that leave the CNS to bring about an effect in a specific organ, smooth muscle, cardiac muscle, or gland. So the autonomic visceral efferents, they actually consist of a 2-neuron chain, a preganglionic neuron and a postganglionic neuron. So let's look at them. The preganglionic neuron typically has its cell body, always has its cell body in either the brainstem or the spinal cord. Therefore, the cell body is in the CNS. The postganglionic neuron is in the PNS. But we need a connection between the preganglionic neuron and the postganglionic neuron, and that connection occurs at what we call an autonomic ganglion, which is located in the PNS as well. So those cell bodies of the postganglionic neurons are typically located in an autonomic ganglion in the PNS, and then they send their axon via an autonomic, those autonomic fibers, efferent fibers, they go to innervate their target organ or tissue. This target organ is also called an effector. So this is just a quick summary of the autonomic nervous system, just the principles of the autonomic nervous system. So we know it's under involuntary control, innervating our organs, cardiac muscle, and glands. We know that we have visceral efferents as well as visceral afferents. And we have two divisions that are always under a dynamic balance. So the sympathetic division, also referred to as the flight or fight division, is dominant in situations that require us to spend energy, situations of stress or excitement, whereas the parasympathetic division conserves energy, restores energy, and is typically referred to the rest and digest division. most visceral organs, but not all, receive both sympathetic and parasympathetic innervations. So these two branches, they have some functional differences, but they also have anatomical differences, and we're going to describe them now. So here we have a schematic view of the sympathetic nervous system. So we have the CNS, and we have the organization of those sympathetic fibers and the target organs. We have a similar schematic for the parasympathetic nervous system and its target organs. So let's start by describing some of those differences. The first difference is when we talk about the outflow of sympathetic or parasympathetic information. So the outflow of sympathetic information only occurs between the T1 to L2 levels of the spinal cord. Therefore, we refer to it as thoracolumbar outflow. The parasympathetic outflow occurs from the brainstem via and then traveling to the periphery via these specific cranial nerves. And we also have outflow from the S2 to S4 sacral levels of the cord. What are some other differences? Well, the ganglia of the sympathetic division, they're highly organized in what we call the sympathetic chain. And you came across the sympathetic chain in body systems. So this chain is also referred to as the paravertebral chain, as it is parallel to the vertebrae. So it is a series of ganglia that extends throughout the whole level, the whole length of the spinal cord. So we have some paravertebral ganglia, and there's also a couple of prevertebral ganglia or collateral ganglia that lie outside the chain. In the case of the parasympathetic nervous system, the ganglia are not highly organized, but rather the ganglia are located near the effector organ. So in this case, it's located near the eyes, near these couple of glands, in the heart here, near the heart, near the lungs, near the liver, near the stomach, and so on and so forth. The last difference is regarding the effect that these systems have. So the sympathetic nervous system has a widespread effect where sympathetic input can actually have an influence on many target organs, whereas the parasympathetic effect is more localized. So if we wish to only constrict our pupils, we're only going to constrict the pupils. Or only induce lacrimation, we can only induce lacrimation. So it's a more localized effect. So now I'm going to describe each of the branches of the ANS in detail, starting with our parasympathetic division, that calming division that allows us to restore energy. So as we were saying, the parasympathetic division is of craniosacral origin. So cranio refers to the origin in the brainstem, and sacral to the origin in the S2 to S4 levels of the sacral cord. So that is where the cell bodies of the preganglionic neurons are going to be located. So either in the brainstem, in those specific cranial nerve nuclei, or in the lateral gray horn of the sacral cord between S2 to S4. The axons of those preganglionic neurons, they leave the brainstem using either cranial nerves 3, 7, 9, or 10. And the axons of the preganglionic neurons from the sacral cord, they leave using the S2 to S4 spinal nerves. Of course, we're going to have a synapse in a ganglion, but I want you to know that acetylcholine is the neurotransmitter secreted by the preganglionic neurons of the parasympathetic division, and it stimulates those postganglionic neurons. And those postganglionic neurons, they send their axons towards the target organ. But actually, acetylcholine is also the neurotransmitter of the postganglionic parasympathetic neurons. And as we can see, we have a 1-to-1 ratio between preganglionic and postganglionic neurons. That means the effects can be localized. One more note. So remember I mentioned that these ganglia, the parasympathetic ganglia, are located near the target organ. Therefore, the preganglionic neurons, those axons of the preganglionic neurons, tend to be quite long, whereas the postganglionic axons tend to be quite short, as the ganglion is near the effector organ. So we already described which cranial nerves include parasympathetic fibers. We have cranial nerve number 3, which goes to the pupil, which have cranial nerve number 7, which goes to some salivary and the lacrimal glands. We know that cranial nerve number 9 specifically goes to the parotid salivary gland, and the vagus nerve is a really important parasympathetic nerve which supplies the thoracic viscera as well as the digestive viscera. So I would like you to take some time and think about, what do you think that these parasympathetic fibers would do to these glands or organs? Would they dilate or constrict the pupil? Would they increase or decrease secretions from these glands? Would they increase or decrease the heart rate? So you may want to pause the video and write your answers in a piece of paper. So I hope you did, but so to come back, so let's think about parasympathetic innervation. So we need to think about situations in which we're allowed to rest, to preserve energy. So we don't need to bring that much light information when we're just resting. So it makes sense that the pupils can constrict. It makes sense that we may be allowed to salivate or tear up in situations in which we're not under stress. The heart rate can decrease. We can constrict our bronchi, so we don't need to bring that much air in. And we're going to increase digestion, so increasing peristalsis, and we can be allowed to digest. So let's look at that parasympathetic division more in detail. So we already said that we have input via the brainstem or the sacral cord. In the brainstem, it travels via those specific cranial nerves that we mentioned. So let's focus a little bit on that, on the anatomy of that vagus nerve. So this is a very long nerve that it leaves from the medulla oblongata and it descends all the way into the thorax and abdomen to provide parasympathetic supply to all of these organs. Okay? So those fibers, those parasympathetic fibers, they form organ plexuses to reach those target organs. Okay? And then they can synapse near the target organ itself. So we already mentioned that the parasympathetic innervation to the heart is going to decrease heart rate. We can constrict those bronchi a little bit. We can allow digestion. So now, what do you think that the S2 to S4 parasympathetic fibers would do to those organs? So those parasympathetic fibers are going to leave as a bunch of pelvic nerves. They're also called the pelvic splanchnic nerves, to innervate the distal large intestine, the kidneys, the bladder, and some reproductive organs. So what do you think? Again, you can pause the video and write your answers in a piece of paper. So we're going to be discussing in a few slides in detail the autonomic control over the bladder and over the distal part of the colon. But it would make sense that if we are resting and digesting, these fibers are going to be allowing defecation and urination. Okay, parasympathetic fibers are also responsible for erection of erectile tissues. So if we go back to those parasympathetic fibers running through the 4 cranial nerves, so remember, I'm not going to go over this in much detail. Remember that those fibers are associated with some specific head and neck ganglia. And after that ganglion, we have postganglionic fibers that piggyback onto divisions of the trigeminal nerve, cranial nerve number 5, to reach the target organs. So we have now finished our discussion about the parasympathetic nervous system, and now I'm going to be talking about the sympathetic nervous system, that arousing system that works mainly under conditions of stress or excitement when we need to spend energy. So let's look at the outflow from the sympathetic division. So the sympathetic division takes origin, those preganglionic neuron takes origin in the T1, between levels of T1 to L2 of the spinal cord. Okay, so we refer to that as thoracolumbar origin. So only between T1 and L2 we're gonna find the cell body in the lateral horn of the cord between those levels. So the preganglionic neuron is going to send its axon towards the PNS, the periphery, and that neuron is carrying with it the neurotransmitter acetylcholine. So acetylcholine is the preganglionic sympathetic neurotransmitter, which is going to it's going to stimulate the postganglionic neuron at the level of the sympathetic chain ganglion. So mainly, most ganglia are organized in the sympathetic chain, but as we'll see, some other ganglia are located, are collateral ganglia, not exactly next to the vertebrae, but around it. However, the postganglionic neurotransmitter, the neurotransmitter carried in the postganglionic neuron, is noradrenaline, also called norepinephrine, for most target organs except the sweat glands. So norepinephrine or noradrenaline is the postganglionic sympathetic neurotransmitter. And this sympathetic division tends to have widespread effects rather than localized effects. The effects are widespread. And there are many reasons for that. So in terms of ratio, there tends to be one preganglionic neuron that can innervate many postganglionic neurons. And I will illustrate that with this slide here. So there are 2 reasons why the sympathetic nervous system has widespread effects. The main one is due to neuronal divergence. So neuronal divergence is when a single neuron can influence the activity of many neurons. So one preganglionic sympathetic fiber can synapse and influence the activity of many postganglionic neurons, and therefore simultaneously stimulate multiple effector organs. The other reason is that we do have some preganglionic sympathetic fibers that leave through the thoracic cord, and they go directly to the adrenal medulla. So they do not synapse with any ganglion, they synapse directly with the adrenal medulla. So what's very interesting about the cells of the adrenal medulla is that they are analogous to postganglionic cells. So we refer to these cells as modified postganglionic cells. And the adrenal medulla synthesizes and releases norepinephrine and epinephrine into the bloodstream. So as soon as these cells of the adrenal medulla are stimulated by the sympathetic preganglionic neurons, we're going to have widespread release of these hormones into the bloodstream, therefore exerting widespread effects. So again, let's talk a little bit more about the sympathetic nervous system. And again, we have a representation or a schematic of those regions of outflow. So between T1 and L2, we have preganglionic fibers. So those preganglionic fibers only exist between those levels, and they leave using the T1 to L2 segmental spinal nerves only. The very reason and the very interesting fact about the sympathetic chain is that it acts as a distribution hub. So we have sympathetic chain ganglia along the entire length of the vertebral column. So these sympathetic chain ganglia, they allow for information to travel beyond T1 and beyond L2. Okay, so input or can only leave, let's say from T1 is the most superior place where we can have preganglionic input, but the sympathetic chain allows for those fibers to travel up so that we still have sympathetic innervation towards structures that are located higher up. For example, structures in the head and face. Again, the lowest area where we can have preganglionic sympathetic input is from L2. But how come we can still innervate organs in the pelvic region? Well, again, because that sympathetic chain allows for some fibers to travel down. Okay? So it is a really clever system that we have. So those preganglionic fibers, they run to those sympathetic chains mainly, and from there they can be distributed. Those postganglionic fibers can be distributed to the different organs of the body. So I mentioned that most fibers run to the sympathetic chain ganglia, but we do have other collateral ganglia that are not part of the sympathetic chain. I don't want you to remember the names of these ganglia. I just want you to know them as prevertebral ganglia or collateral ganglia. And we're going to talk about why they exist. But for now, I would like you to again pause the video and in a piece of paper write, what do you think that sympathetic fibers are going to be doing to these target organs? So if you're back with me, remember that the sympathetic nervous system is necessary for conditions in which we need to spend energy. We need to get ready for situations of stress or excitement, running a marathon or writing our final exams, okay? So, or running in a dark forest from a snake. Well, I don't even know if it could be, depending on the forest and depending where you are. Anyway, so normally we would like to bring as much light as we can into our pupils as possible. Therefore, we would dilate the pupils. We would inhibit secretions from the salivary and lacrimal glands, as we do not want to be salivating or lacrimating in situations of stress. Heart rate, what's going to happen to the heart? Well, the heart rate's going to go up, okay? The bronchi are going to dilate because we need to start breathing more heavily to try and bring more oxygen in situations of more stress and where we need more energy. What's going to happen with digestion? Well, we need to inhibit digestion. We do not want to be digesting when we're running a marathon or writing an exam. the liver is gonna get ready to secrete, to convert, to give us readily available source of glucose. Okay. Again, what's gonna go on with the intestines? Well, peristalsis and digestion are gonna be inhibited. We do not want to be defecating or urinating when we are in situations of stress or excitement. Okay. So some of you, or all of you, I hope, had the chance to see the sympathetic trunk, or also referred to as the sympathetic chain, during body systems in our DR. And hopefully you can remember that it is an interconnected chain of ganglia located on either side of the entire vertebral column. So that's why we refer to them as the paravertebral ganglia, because they're parallel to the vertebral column. So here we have a right view, and the sympathetic chain is highlighted for us. So we can see that at each level we have some ganglia. And in this left view, we can see that sympathetic chain as well. So these are the sites of synapse between the sympathetic pre- and postganglionic neurons. And as I mentioned earlier, they permit preganglionic fibers to ascend or descend beyond T1 and L2. Okay? So now what we're going to be talking about are some outflow options, some sympathetic outflow options, because the preganglionic neuron can actually take a couple of different routes in its path to finding a ganglion where it can synapse. So the first option, indicated by the number 1, is the most straightforward option, in which the preganglionic neuron leaves the cord, synapses right away near the level of entry with a ganglion in the sympathetic trunk, so a paravertebral ganglion, and from there we have a postganglionic neuron that is going to travel to its target organ. So those postganglionic neurons go mainly to organs in the thorax and the glands and smooth muscle near the body wall. So for example, for making us sweat. So just to summarize, that preganglionic neuron leaves the cord, synapses right away at the level of entry with the postganglionic neuron, and then the postganglionic neuron goes to organs in the thorax or skin and body wall. Let's look at the second option. So what happens in the second option is that the preganglionic neuron leaves the cord, but it completely bypasses the sympathetic ganglia. So we do not have a synapse in the sympathetic ganglia, because that preganglionic neuron is gonna find a synapse in one of those collateral ganglia, in one of those prevertebral ganglia that are outside the sympathetic trunk. Okay, so one of those collateral ganglia. So from that collateral ganglion, then we have a postganglionic neuron that goes to a specific organ, and these tend to be organs in the abdomen or in the pelvis. Lastly, our third option is when the preganglionic neuron leaves the cord. So these tend to be either really close to T1 or really close to L2. So the preganglionic leaves the cord, does not synapse in the sympathetic chain ganglia, but ascends a couple of levels to synapse with another ganglion in the sympathetic chain closer to that target organ. So for instance, if we're going towards organs of the head and neck, it makes sense that the preganglionic neuron wants to go towards those ganglia in the chain higher up, synapsing and then sending a postganglionic neuron to a target organ in the head and neck. And it works similarly to those lower organs in the pelvis, where the preganglionic neuron is going to leave those lower levels of the lumbar cord, so around L2, bypass the sympathetic chain, descend in the chain a couple of levels to find a ganglion lower down, and from there synapse and go to a target organ in the pelvis. Okay? So those are the different ways that the preganglionic sympathetic fiber can leave the cord and travel on its way to reach a ganglion. So I want to tell you about some structures that are associated with our sympathetic division. So here we have our typical cross-section of through the spinal cord. The fact that we have this little lateral gray horn tells us we are in that thoracic cord and maybe L1, L2. And we have the spinal nerves. So any spinal nerve at any level, it has a dorsal root associated with a dorsal root ganglion and a ventral root, and we also have dorsal and ventral rami. However, with the sympathetic division, we have additional structures. So we have, of course, the sympathetic ganglion in that sympathetic chain between T1 and L2, but we also have some additional structures that are referred to as the rami communicantes. So essentially what this means is communicating branches. So rami means branches and communicantes means communicating. So we have a white ramus communicans and a gray ramus communicans. One key difference between these two is that the reason the white ramus communicans is referred to as white is because myelinated fibers run in that branch. Okay, so it appears white. But in the gray ramus communicans, only non-myelinated fibers run there, so it appears gray. So the preganglionic neuron of the sympathetic branch, we already said it has its cell body in that lateral gray horn, only between T1 and L2. We already mentioned how that preganglionic neuron leaves via that ventral root. But what's very interesting is that the axon of that preganglionic neuron is actually myelinated, and it's going to use the white ramus communicans to leave. Okay, and now depending in one of those pathways that we talked about from the previous slide, we can have a couple of different things happening. So that fiber can, for instance, completely bypass that sympathetic ganglion, as we mentioned earlier on. Or if we have a synapse right there with a postganglionic sympathetic neuron, let's say we have a little connection there, that postganglionic sympathetic neuron is non-myelinated, and it's going to use the gray ramus communicans to rejoin that spinal nerve. Okay, so we have these rami communicantes associated with the sympathetic division, and they simply provide a route for those pre- and postganglionic axons to travel. Okay, so remember the reason the white ramus communicantes is called white is because preganglionic myelinated axons run there, whereas the gray ramus communicans contains postganglionic myelinated axons. So with this schematic, I'm just going to summarize those outflow options, those sympathetic outflow options. So let's look at an example. So just here we have a cell body of a preganglionic sympathetic neuron. So that preganglionic sympathetic neuron leaves via that ventral route, and we can see how it's traveling in that wide ramus communicans. So in this first option, it's immediately going to synapse at that sympathetic chain ganglion with a postganglionic neuron. Okay, and it can either go to the cardiopulmonary system or use that gray ramus communicans to rejoin the spinal nerve and travel. Those postganglionic fibers can travel to the body wall via those spinal nerves. The second option we talked about is when the preganglionic neuron leaves the cord. So here we have that cell body of the preganglionic neuron, but it completely bypasses the sympathetic chain, so it does not synapse in the sympathetic chain because it wants to find a prevertebral ganglion, okay, a collateral ganglion, where it's going to synapse and go, and the postganglionic neuron is going to travel to those digestive organs. The third option, which we've revealed up here, is when the preganglionic neuron, so it leaves via that ventral route, and instead of synapsing in the chain, it's going to go up or down a couple of levels to either innervate structures in the head and neck, if it's the case of going higher up, further up in the chain, or in the case of going lower down in the chain, innervating pelvic organs. So what we're going to do now is discuss in a little bit more detail some of the autonomic control of a couple of organs and structures. So in this slide, I'm going to tell you about the autonomic control of heart rate and respiratory volume. So let's start by looking at the sympathetic control of heart rate. So those sympathetic fibers, those preganglionic fibers, they leave the cord from T2 to T4 levels of the thoracic cord, okay? And those preganglionic neurons are going to find a postganglionic neuron in different levels of the sympathetic chain. So from the sympathetic chain, postganglionic neurons are going to travel towards the heart in what we call these cardiac nerves or cardiac plexuses. Okay? So we have short sympathetic preganglionic neurons and long sympathetic postganglionic neurons. And as we've mentioned earlier, the sympathetic division will stimulate heart contractions to increase blood flow in situations where we need to prepare for stress and spend energy. Okay? Since we're here, let's talk about the sympathetic supply to the lungs. So again, we have some input from these T2 to 4 levels of the cord, and we're gonna have postganglionic neurons coming from other sympathetic chain ganglia a little bit down the line, so closer to the lungs. So those postganglionic neurons, they leave via different kinds of pulmonary plexuses, pulmonary nerves to the lungs, okay? And the sympathetic effect on the lungs is to induce dilation of the bronchi so that we can bring more air and more oxygen, okay? So those are the sympathetic effects on these organs. Let's look now at the parasympathetic effects. So we know that the chief parasympathetic nerve to the thoracic organs is supplied by the vagus nerve, okay? So we know that the vagus nerve leaves from the level of the medulla oblongata in the brainstem. So an associated parasympathetic nucleus in the brainstem is going to contain those preganglionic parasympathetic cell bodies, and the axons of those preganglionic neurons are going to leave via that long vagus nerve and descend to the thorax to innervate those organs. So we're gonna find a synapse at the level of the target organ, be with the postganglionic neuron, as well as the same story for the lungs. So what are the parasympathetic effects on the heart? Well, we're gonna inhibit contractions of the heart, so the heart rate can lower a little bit, and we can induce constriction of the bronchi, so less air is gonna be flowing into our lungs in situations where we are allowed to relax. Okay, so I have described those visceral efferent fibers of the sympathetic division and the parasympathetic division that bring about a specific effect. So what about those afferent fibers that monitor the state of some of those internal organs? Well, one example that I'd like to give you is, remember we talked about in the cranial nerves lecture that we have some structures really close to that bifurcation of the internal and the bifurcation between the internal and external common carotid arteries. We have some structures referred to as the carotid body and the carotid sinus, which are constantly monitoring information on blood pressure and information on the levels of oxygen. So actually, visceral sensory afferent fibers can travel via that glossopharyngeal nerve towards the medulla oblongata, bringing information about the state of oxygen in the blood and the blood pressure. And we can have some integration going on here that can tell the brainstem whether blood pressure is too high. We can lower it down using that vagus nerve, for example. Let's look now at the autonomic control of micturition, which is urinating. And this is a very, very cool example. I think cool is an appropriate word, because the bladder actually receives sympathetic innervation, parasympathetic innervation, as well as somatic innervation. So let me tell you what's going on. There's actually a lot going on, but let's start by talking about the somatic innervation, because the somatic innervation, it actually forms the default state. So the default state in the adult is that our external urethral sphincter is constantly under constant contraction. Okay, that's why we're not really peeing all the time. It's constantly being contracted by the pudendal nerve. Okay, so that is the default state. So the pudendal nerve provides contraction to the external urethral sphincter, which closes the external urethral opening to prevent urine from leaving. Okay, so you may be aware that urinating is actually voluntary. So when you feel like you want to urinate and you're allowed to, you're not in a bus, or you're not delivering a lecture or taking the lecture, for instance, then you can hold it in. However, when you are allowed to go and urinate, then you voluntarily, it is actually your brain that voluntarily sends instructions all the way to the pudendal nerve, which takes origin in the sacral cord, to relax that external urethral sphincter. Sorry, this iPad sometimes. So when we want to urinate, the external urethral sphincter relaxes. Okay? Relaxes. this is the reason this is one of the reasons what's going on during infants, not during infants, during infancy. Infants really cannot hold their urine because they do not have acquired that voluntary contraction of the external urethral sphincter. So let me explain what happens in terms of the other innervations. So let's start by looking at the sympathetic innervation, which prevents urinating. It prevents voiding. So between these levels of the cord, we have that sympathetic input. This is one of the cases where the preganglionic neuron descends all the way to synapse with a ganglion lower down in the chain. And then we send some postganglionic neurons towards the bladder. They travel via those hypogastric plexuses, but you don't have to remember that name. What I do want you to remember is the structure of the bladder. So hopefully you know this from year 1 body systems. It's a balloon-shaped structure, and the muscle of the bladder is called the detrusor muscle. So the sympathetic nervous system, it relaxes that detrusor. That's what's meant by this negative sign. It relaxes the detrusor muscle. Okay, so it does not contract it relaxes the detrusor muscle. And it also contracts, that's what's meant by that positive sign, contracts the internal urethral sphincter. So by relaxing the detrusor muscle and contracting that internal urethral sphincter, we're allowing filling the bladder with urine. So the detrusor muscle can expand and allow filling of urine. And that urine is not going to go anywhere because that internal urethral opening is closed by the internal urethral sphincter. So what happens when we want to urinate, when we want to void? So there are some stretch receptors in the bladder itself which sense the level of distension of that muscle, of that detrusor muscle. So when the level of urine reaches a certain level, the impulses via those visceral afferent fibers, we have increased impulses from those sensory fibers that sense the stretch being generated by lots of urine. So those afferent fibers, they reach the cord via the typical pathway. Okay, and what we have here is pretty much a reflex arc. Okay, so if we wish to urinate, the parasympathetic nervous system, which we know leaves from the sacral regions between S2, 3, and 4, those preganglionic parasympathetic fibers, what they're going to do is that so the preganglionic parasympathetic fibers leave, they reach a ganglion near the target organ, and they're going to, in this case, contract the detrusor muscle to compress that muscle and allow urine to leave, and they're gonna relax the internal urethral sphincter so that urine can start leaving the bladder. So we're compressing that balloon and we're opening that internal urethral sphincter. However, we are not going to urinate until we voluntarily decide to do it, until we voluntarily tell our pudendal nerve to relax the external urethral sphincter. Okay? So until we tell our pudendal nerve, until our brain sends signals telling the pudendal nerve to relax, then we can actually urinate. So in infants, what's going on is it's always a reflexive bladder. So, sorry. I just want to find the eraser, but the iPad doesn't seem to be allowing me to do that today. So I mentioned earlier that infants don't have yet developed that voluntary contraction via the external urethral sphincter. So in infants, what's going on is always this reflex arc here, a reflexive bladder, where as soon as there's enough distension of the bladder being sent by those sensory fibers, as soon as those signals are sent to the spinal cord, it is the parasympathetic nerves that immediately send signals to contract the detrusor and relax the internal urethral sphincter. So let's look now at the autonomic control of defecation. So this is quite similar. So the sympathetic inputs leave from the lumbar cord, and again they bypass the sympathetic trunk to find a collateral ganglion. You don't need to know the name of that ganglion. And then we have postganglionic sympathetic fibers that go towards the distal colon and rectum. So again, during situations of stress, we're going to prevent peristalsis and emptying of the rectum. What's going on with the parasympathetic innervation? Well, those preganglionic parasympathetic fibers, they leave via the S2, 3, and 4 levels of the sacral cord. And in situations where we're allowed to rest and digest, those innervations going to the distal colon and rectum are going to induce peristalsis and emptying of the rectum. However, we also have our pudendal nerve, and as you may be aware, defecating is also under voluntary control. So the default state is that the pudendal nerve is always inducing contraction of our external anal sphincter, preventing defecation most of the time. Okay? So when we voluntarily want to go to the toilet and empty the contents of the colon and rectum, we are going to have a voluntary inhibition of the contraction of the external anal sphincter. Okay? And the parasympathetic nervous system is going to take over, induce peristalsis and emptying of the rectum. So again, we have some stretch receptors in the rectum that sense the distension of the rectum by increased levels of stool. And again, this voluntarily control is not present in infants. So the autonomic control of pupillary and accommodation reflexes is going to be discussed in the visual pathway lecture. So I thought I'd let you know about that. So let's look at some of the injuries that we can have in terms of the autonomic, that would affect the autonomic nervous system or involve the autonomic nervous system. So I'll just tell you a couple of examples. So some sympathetic injuries. So one example that we've seen before is Horner syndrome, and we talked about Horner syndrome during clinical cases number 1, in which if we have any injury to the sympathetic supply to the head and neck, so for example, injury to the sympathetic trunk at the level of the cervical vertebrae, or injury to the upper thoracic segments of the cord. So if we interrupt that sympathetic supply to the head and neck, we get a condition called Horner syndrome, which is characterized by miosis, ptosis, lack of sweating, and redness. Okay, so you can go back to clinical cases number 1, and there's a nice slide about Horner syndrome. So if we have injuries to the cord or the coda equina, we're going to have disruption of the sympathetic control to the bladder. Let's talk about parasympathetic injuries. So again, there's multiple ways there can be parasympathetic injuries. So for example, injuries to those parasympathetic, those nerves that carry parasympathetic fibers, so 3, 7, 9, or 10. So we can have failure of the pupils to constrict with an injury to cranial nerve 3. We can have problems with salivation or lacrimation with injuries to cranial nerve 7, and multiple problems with innervations of our thoracic or abdominal viscera if there's injuries to the vagus nerve. In terms of injuries to the sacral cord or coda equina, well, again, we're going to have disruption to the bladder, the rectum, as well as disrupted sexual functions. So we can predict a little bit what can happen with different kinds of injuries to the cord in terms of urination, for example. So if the lesion in the cord is above S2, 3, and 4, that means the parasympathetic supply to the bladder would still be preserved. However, we would lose the sympathetic control to the bladder. So for example, if we have an injury at the level of L1, L2, or the roots. So what we would have in that case would be a case of a reflexive bladder, similar to what happens in infants. And the reason for that is because we are going to lose the descending. So if the lesion is, let's say, at L1, L2 of the cord, we're going to lose those voluntary descending fibers that supply the external urethral sphincter. Okay, so as soon as we have those stretch fibers sensing distension of the bladder, immediately those S2, 3, 4 parasympathetic fibers are going to take over and induce urination. We have a loss of that voluntary control. Okay. What would happen if we have an injury to the sacral cord, so disrupting S2, 3, 4, or we sever the coda equina? Well, in that case, we would lose both parasympathetic supply, and we are also going to lose that voluntary control to the bladder. We would also lose those sensory fibers carrying information from stretch. So in such a case, the bladder can become flaccid, so the detrusor muscle would continuously be relaxed. It would fill to capacity, okay, and we would have continuous dribbling of urine in that situation. Last but not least, I did mention earlier on that the hypothalamus was quite important because it rostrally regulates autonomic functions. So let me briefly discuss the anatomy of the hypothalamus. So we've mentioned already that it is part of the diencephalon. It is, let me go back. I don't know what happened. I touched the slide in the wrong way. So the hypothalamus is part of the diencephalon, so separated by the thalamus by this hypothalamic sulcus. It is bordered anteriorly by the lamina terminalis, ventrally by the optic chiasma. We know that we have the infundibulum, which connects the pituitary gland to the hypothalamus. We have those mammillary bodies as well, and bordered inferiorly and posteriorly by the brainstem. So the hypothalamus, I think it's quite underrated because even though it only forms about 0.3% of our total brain, it has many important functions that are essential for life. So it controls and influences the activity of the autonomic nervous system. So some structures in the hypothalamus are going to induce sympathetic effects. Some structures in the hypothalamus are going to induce parasympathetic effects. The hypothalamus also has a central role in our neuroendocrine function via connections with the pituitary, as it releases a couple of hormones, and it also controls the synthesis of other hormones. And the hypothalamus also has very important connections with the limbic system. The hypothalamus itself consists of a series of nuclei, so similar to the thalamus that we had many nuclei. We have many nuclei of the hypothalamus, but we don't have to remember those nuclei. So this brings me to, I believe, the last slide of today, which is where we're going to be looking at those levels of autonomic control. So the hypothalamus, it exerts overall control and regulation of our ANS. So the hypothalamus can influence our brainstem centers, some of which are important for cardiovascular control, respiratory control, as well as swallowing. And it can also influence the spinal cord centers in the thoracic cord for sympathetic, so the thoracolumbar cord for sympathetic input, and the sacral cord for parasympathetic input. So those spinal cord centers that influence urination, defecation, erection, ejaculation, etc. Okay, so the hypothalamus can have influence onto these other levels. Furthermore, we also have, remember, those autonomic afferents that are constantly relaying information about the internal state of the body back to the CNS and eventually back to the hypothalamus. So we can have information going the opposite way as well. So we have that visceral afferent input relaying information to the hypothalamus, which can then decide what's the next step. Whether heart rate is too low and we need to increase it, whether we need to increase body temperature, whether we need to increase the release of different hormones that are going to cause vasoconstriction, for example, etc. Last but not least, and this is one of my favorite aspects, is that all of this is actually regulated as well by the cerebrum. So our cerebrum can play very important control in the way we regulate our internal environment. So importantly, one part of the cerebrum is the limbic system, which is important for our emotional state and memory. So you may have realized that your memories can trigger powerful autonomic effects in your body. So for example, if you remember right now your favorite takeaway in Leeds or wherever in the world you are, you might start salivating. How come? Well, that is your memory, your cerebrum having important connections with the hypothalamus, controlling those important centers for salivation. Another example, if you remember a situation in which you were very happy and highly upbeat, a really nice memory, your heart rate might start increasing without there being any necessary source of stress or need to spend energy. If we are feeling joyful, if we are feeling sad, if we are feeling depressed, all of those emotional responses can actually influence upon the activity of the ANS. And that is really due to important connections with the hypothalamus. Okay? So we will talk about the limbic system later on. So this is the summary of today's lecture. So you should be able to distinguish by this point between the somatic and the autonomic nervous systems. You should be able to describe the anatomy of the ANS, compare the parasympathetic and sympathetic divisions. You should be able to tell us the organization of autonomic efferents using the terms preganglionic neuron, autonomic ganglion, postganglionic neuron, and effector organ. You should be able to describe the organization of the parasympathetic nervous system and describe those cranial nerves that carry parasympathetic fibers. You should also be able to describe the organization of the sympathetic nervous system. And you should briefly be able to describe the autonomic functions, including heart rate, respiratory volume, urination, defecation, and pupillary reflexes, which, as I mentioned, will be discussed later on. I will leave you with this last slide, which I find quite funny, and